All right, well, we might as well get started. Um, this is kind of an unusual way of doing this, I guess, but Miss Crystal has said everything are just wonderful. I think it's gonna go off great. One of our past professors in entomology, applied entomology, Garden City, Dr. Larry Buschman, has had I think probably about a lifelong interest in lightning bugs, fireflies, and he's wanting to share this information with us. He's evidently had some health problems, so he couldn't make it here, as you can see, but he kindly sent these little pamphlets from the Kansas School of Naturalists in Florida State, Fireflies in Kansas, by Larry Bushman. So, without further ado, Larry, I'll let you go for it. Thank you.
Okay, thank you. And I just wanted to show my great granddaughter. She got excited with insects and showed her mom that she identified it as a cicada. So this is now 15 years after I retired. I didn't realize it was quite that long, and that's why there aren't that many people that remember me in the department. When I come to the department, I only see one or two of these individuals that I can remember. So, but my 15 years have been, I've had good 15 years, and my great grandkids are getting to know the fireflies, and they know exactly where to find them in the house. And my grandkids are settling into their careers. That was one of my motivations when we retired. We needed to support them. The family was falling apart. And Rose and I have done a number of short-term mission and service projects around the world. And, that's been good, but that also is not happening anymore. Our health isn't allowing it. I have completed quite a number of writing projects in fireflies. And I sent some of these pamphlets that should be there. You pick one up if you really want one. The pamphlet actually gives a lot of the biology of the fireflies. And then there are these tables with the fireflies of Kansas for identifying. It's a field guide. And so that's the big contribution that I've been working on. I've also done some life history or family history, and actually published in another profession, missiology. So that gets to be a different enterprise when you try and do that. And then we also lived in Colorado in the mountains for a while because my wife grew up in the mountains in British Columbia, and she was just always gravitating toward the mountains here when living in Kansas. And so we went there and lived for 9 years, and that kind of filled her need there. So now we're back in Heston. So I wanted to talk about fireflies and let you know that I'm doing it for the fun of it. Several years before I retired, I was part of a search committee, and we were looking to fill a pest management position. And we had a number of very good candidates to work with, and we had to find a way of identifying, okay, which of these candidates do we really want? And so I pointed out that, hey, there are two of the candidates were pest management trained, and two of them were trained as ecology people. They both work as pest management positions. But I told him my experience before coming to Kansas State University, I had been an ecologist. And at Kansas State University, I converted to working as a pest management researcher. And that my experience was that as an ecologist, I got to choose what topic I wanted to do my research on. But as a pest management researcher, I had to study whatever the farmers were working with or dealing with. And there's a little bit of a difference there. So Now, when I got to retirement, I had to make that decision again. Do I continue to work with the pest insects or do I work with fireflies? I got invited to see the synchronous fireflies in Tennessee. And I tell you, if you guys have not seen the synchronous fireflies, it's impressive. You need to get there. That experience reminded me just how much fun it was to be working with fireflies. And so I reverted to going back to being an ecologist and working on fireflies. So I'm doing entomology for the fun of it again. So in this presentation, I'd like to cover kind of three topics. They kind of lead from one to the other. First, there's the rearing of the fireflies, and then the courtship flash dialogue, and then the biological species concept that grows out of it. And so I'll try to minimize the presentation in each one because it's a big, each one of these is a major topic. This is what the Photurus firefly larvae look like. And one of the things that I wanted to point out, notice that this firefly on the right, the head is very small and narrow. That's because he telescopes that head in and out from under that pronotum. And that's one of the characteristics of the lampreyrids. The head is always retracted under the pronotum. Most other insects have it attached more or less like a hinge or some other form of attachment. So rearing the new insect, normally you have two obstacles. What do you feed them? And then what do you do to stimulate them to produce adults? And because you can feed them for all you're worth and sometimes they'll never pupate unless you give them the right signal. During my PhD project, I had identified what Photurus larvae were eating, but I hadn't actually gone to rearing them because I needed to graduate. And so I left that to the side. So now, and I also knew how to stimulate them to pupate. So now in retirement, I had a little more time. And so I decided I really wanted to do that. And it also seemed like the kind of a project that I could do into retirement in case, I got a little bit infirm or something like that. I probably would be able to do the rearing stuff a little bit better than some of the other projects. So my rearing adult firefly goes into the large jar. I pack a little bit of soil on one side that the larvae or the adults, females lay their eggs there and they hatch. And then I feed them earthworms that I get from the fish bait shop and cut them up and add the earthworm to the section, and the larvae feed on the earthworm. And this picture actually shows many larvae in one situation. Actually, I have to separate them into individual jars. And so on the right, you can see all the individual jars because they are predators on each other. And so I have to rear them as individual larvae. And then I also have to show this because it's a photoperiod chamber. The upper box is set to go 14 hours of light and 10 hours of darkness. And the lower chamber is set to go 10 hours of light and 14 hours of darkness. The long photoperiod simulates summer, and the short photoperiod simulates winter. And this is just one of the projects that I've done. And you'll notice these bars are showing the percentage of adults that are produced. And I put in 3 different treatments. And the first treatment is larvae went into the long photoperiod and continuous photoperiod. I didn't change the photoperiod over time. And you'll notice that no adults were produced. The same thing happens with putting them in continuous short photoperiod. They won't produce any adults. It's only when they've been in the short photoperiod and I switch them over to long photoperiod, that switch is the trigger that gets them to go forward. And so that's critical in this. And so one of the things that I'm doing is trying to measure how long it takes and that kind of thing. But notice that the larvae from Fort Collins, Colorado, never did pupate. But when I kept rearing them, this is the first winter. When I reared them through the second winter, then I did get some adults. So I'm dealing with a diapause here that's induced by the photoperiod, but it's different from most other insects. Most other insects go into diapause and they get lethargic or become inactive, and they to survive the winter. But these guys, the larvae, continue to be active. They feed, they grow, and the only way I can tell that they're in diapause is they refuse to go to pupate. And apparently the diapause here functions in timing the adult emergence in spring. It's all about getting male and female to emerge at the same time so that they can mate and produce the next generation. There is also estivation going on, and they can be either diapause or non-diapause larvae. They just go into the soil and wait until conditions become more favorable. This is kind of my working hypothesis, and I'd really like to know if there's other insects that anybody knows about that would be worth my looking into, because in this case, the three types of larvae I'm finding. The first grow real rapidly in about 2 or 3 months, and I can put them into the long photoperiod and they produce the adult almost like clockwork in about 60 days. There's always 10 to 20% hangover, and that need to go to the second winter. And so, that seems to be an interesting thing, and I don't think it's by chance. I think it's an adaptation that some of the larvae hang on for the second generation. Another group of larvae are kind of intermediate growers at first, and when I put them into the long photoperiod, they will produce adults, but it takes quite a bit longer. But most of them have to go to the second year, and then they will continue to grow. And sometimes they go to the third year. And then I have some larvae that are so slow that they don't even grow very much during the first year. And then it takes 2 or 3 years, and but so far I haven't gotten anything that actually produces pupa beyond 3 years. The survival rate is kind of low. Okay, courtship. Once I produced adults, then I needed to, I was watching to see whether the adults were producing a flash dialogue that I could recognize, and I could tell that they were actually producing a dialogue between the male and the female, but it was hard to figure out exactly what was happening. So I recorded it on video and then did a frame-by-frame analysis. And so that's the way I've been able to record the courtship dialogue for fireflies for the first time. And in the field, occasionally we observe the dialogue, but it happens so fast that you can't really describe it. And so this is now a new method that I can do, describe the flash dialogue of the fireflies. This is what the dialogue looks like for Photurus quadrifulgens. And these are male flashes up here. And the female responses in green, notice that they're short flashes and kind of choppy. Here's another species, Photurus tremulans, and the male starts flashing and then the female chimes in at it on a different rhythm. And so they go on for a while. So it's a different kind of a flash exchange. So in order to prove that I really had the flash dialogue for the female, I went to see if I could find somebody that would help me develop a decoy that would produce the female response in accurately. And KSU didn't help me, but I went to KU, and they helped build a firefly simulator with 2 buttons here and so I set the timing up for those 2 species, and I can use 1 button to produce the female response flash. And in the field, these response decoys just work tremendously well. I can attract fireflies from up 50, 60 feet in the tree, and they'll come down to the floor and land right on the LED. So using the decoy as an aid, it helps with identifying the fireflies. I'm working also to develop a library of different species. I think there's about 15, 16 species that I can produce right now. And so the decoys are helpful, and then I can use it to identify the fireflies. And here's an example of how I've used the decoy. In Colorado, I had a situation where we had flashes that were 1 to 5 seconds apart, and that's a large spread. And but it's continuous. Normally, if you have 2 species involved, you'll have a peak here and a peak there. But in this case, it was different. And now, if I separated the flashing by early evening and late evening, I could separate the early evening were shorter and the late evening were much longer. But when I took the decoy out there, I found that short flasher responded to the trim response flash, and the long flashers were attracted to the fast multiple flash decoy. So they're responding to different decoys, and so at this point I'm convinced that there are 2 species involved in this population. And so I call them the Colorado short flasher and Colorado long flasher. In Kansas, it gets a little bit more complicated because there are more species here. But that's the kind of thing that I've been working with. So the biological species concept, I was working with Jim Lloyd at the University of Florida, and he identified that fireflies are using the flash dialogue to identify themselves. And so if we can actually use the flash dialogue to identify the fireflies, because that's the way the fireflies themselves identify themselves. And he actually demonstrated that in the genus Photinus, this works just super well, no problem. But then he spent 50 years trying to make it work with the Photurus fireflies. And the problem is that with the Photurus, there are several different flash patterns. Each flash pattern has a different function, and whereas Photinus only has a single flash. And secondly, we need to know the mating dialogue. And in Photinus, he had the information about both the male flash and the female flash. In the Photurus, he didn't have that. All he had was the male flash. And so now that I have been able to collect the the mating dialogue information, I'm working to catalog the different mating flash dialogues for the Photurus fireflies. Now, using DNA, that's the new high-powered technology. And I've tried several times to find individuals that would work with me to do some DNA work. However, the CO1 does not seem to have enough information to really work with these Photurus in terms of identifying species. And many people have failed to find consistent signals. And it appears that the DNA also seems to be degrade more rapidly in fireflies than in other insects. So there are complications. I'm not going to be able to solve that DNA problem with the fireflies. So the publication situation is that I have a number of new species. I can't publish the rearing or the flash dialogue information that I have because I don't have the species name. And I can't publish the species descriptions because they're based on the rearing and the dialogue information. It's kind of a catch-22. And then I have reviewers that will tell me, well, send your specimens to an expert. And I'm saying, if I'm not the expert, who is the expert? I could fake it and just slap a meaningless name on the population. So, in the past, in the literature, you have Photurus pennsylvanica and Photurus versicolor. But, the minute you do that, that's the same as saying Photurus species. You're not really saying anything. And also, it confuses the literature because there actually is a Photurus pennsylvanica and there is a Photurus versicolor. It's a specific species. So, I've got a catch-22 there. There's so much information. I tried putting together the manuscript for the fireflies in Colorado, which is just a few species, and it got to 60 pages, and I still hadn't gotten to the tables and figures and so forth. There was no way that I was going to get it into a simple manuscript. So I'm still working on how to get that put together. And then the reviewers are going to say, where's the DNA data? Well, I don't think the DNA is going to tell us very much, and it's not the kind of thing I can do either. So I may just have to self-publish without going through referees. So I'm going to turn it back over if maybe I can answer some questions.
Any questions? Larry, I have a question about the flashing. Do they do that prior to mating, after mating, during mating? Is it an attractant? You said it was an attractant for mating purposes. And along with that, do they produce a pheromone? Do you know?
Okay, firefly. Apparently, the primitive situation was for fireflies that they had pheromones. And then the bioluminescence apparently was functioning mostly as a warning to predators that to identify that they're not tasty. And so then that became a second useful thing. And so when they moved into the night, then they could use the flashing for mating. But I need to understand your question. What was the first part of the question?
Do they flash until they find a mate, and then does the mated female quit flashing, or the mated male? Do they mate more?
No, they keep on flashing because it still functions, as an aposematic signal. So they're flashing all their life. But when I'm rearing them, I find that one of my main problems is that I have to have a male and female available at the same time, and they're both sexually uh, active. And the female has to be between the age of 3 to 7 days. The male can be older than that, but rearing them out and getting them to be present at the same time is not that easy to do. So I usually have to have 30 or 40 fireflies that I'm working with, and then I have a decent chance of having male and female at the same time, so that they I can see the flash dialogue between the male and the female.
Just shout that. Tom.
So Larry,
Yes.
do both male and female flash, or I wasn't paying attention maybe, or do they have? I thought it was just the male.
Well, it depends on which group of lampyrids we're dealing with.
Uh-huh.
In the pamphlet, I mentioned that we have 3 groups. So you have the flashing fireflies, you have the glowing fireflies, and you have the dark fireflies. The dark fireflies don't have flashing, are not luminescent as adults. Okay, the glowing fireflies, normally it's the female that glows. The male may have a little bit of a glow or may be completely dark. So, you can have a situation where the male is dark, but the female is glowing. And, he flies around looking for that glowing female. And then you have the flashing fireflies where both male and female are flashing, and they use a signal system, a flash answer signal system to find each other.
And is there somehow sexual selection where a female will choose a male with certain flashing, or a male will?
Yes, a number of people have worked with that, with the Photinus fireflies, not with the Photurus. And there appears to be sexual selection. I am not. Okay, it's not my thing, so I haven't gone into it. I hesitate to say I don't believe it, but it's there, and they have shown it, and so okay, let's leave it at that.
Oh, I was going to ask about sperm competition, but we can't go that far.
Well, with the Photinus, you do have sperm competition, because you can actually open the female and see if there's 1, 2, or 3 spermatophores present.
Right.
But in Photurus, it appears to me they mate only once.
Uh-huh.
So, the level of competition for sperm competition, I think that would be relatively minor, simply because they're only mating once.
And does she lay the eggs close by where they mate, or?
In Photurus, the female flies around and lays single eggs all around in the environment. So she's scattering them all over the place. Now in Photinus, they tend to lay groups of eggs in a group, in a clump.
Right.
And so then there would be a little bit more room for that kind of behavior.
Thank you.
So I have one more question, going back to the flashing. They flash all their life, but the female only mates once?
One group.
Yes, but remember, the flashing is not sexual all their lives. The primary function of bioluminescence appears to be the warning signal to the males or to any predators, to let them know that, hey, I'm an insect that has toxic chemicals. You're not going to want to eat me.
Okay.
And so that's so they're flashing for that purpose. And then Photurus actually appear to have separate flashes that they use in courtship. And then they have other flashes that they use as aposematic flashes. In Photinus, that's the same flash. They're using the same flash for both functions.
I was just curious. Correct me if I'm wrong, but I heard that some firefly species mimic other firefly species to like lure them like as a predation tactic. So have you encountered that in your research at all? And does that make it harder to like differentiate like species based on flashing patterns?
Yes, and that's a very interesting question. It's a little bit more complicated than that. But Jim Lloyd, the guy that has spent 50 years working on Photurus fireflies, spent all his life, and he emphasized these things. That was one of his early discoveries as a graduate student. And there definitely is a decoy function that the female has and draws in other species to, and prey on them. The female Photurus actually will feed on other fireflies, and that's one of the few adults that will actually do much feeding. However, I am finding that it's not as widespread as he was thinking, and whatever. I find that kind of behavior in eastern Kansas. I'm here in, close to Newton, and from Newton west, I almost never see this kind of predation, the aggressive mimicry. But when I go to eastern Kansas, go to Lawrence or anywhere in the eastern part of the country, there's aggressive mimicry going on. I have not been able to study it and differentiate it from the actual mating, because it looks to me like they are using the same signal for responding to their own Photurus predator or male that they're using for for predation. But I can't rear these other fireflies, so I never have the other fireflies to test the aggressive mimicry stuff. So somebody else is going to have to finish dealing with that. But it is there. There is aggressive mimicry, and if and I've always thought if I was a girl, Photurus fireflies would be the perfect insect to work with, because they really have it over on the males.
That's true with every.
So earlier, just you said warning lightning, so warning flash, which is you said the most frequently found species. So what are the major predators? And I wonder, any of predator is going to be having hard time to locate those female, and it's like what kind of predator is a target for that warning flashing?
Yeah, that's one of the issues that's kind of hard to deal with, especially with this whole question of aggressive mimicry, because much of the behavior is done in a prophylactic sense. You're doing it ahead of time. You don't even know that the predator is there to see what the warning signal that you're giving. So I don't know. We think that predators, they're bats and birds and various insects. But things like spiders are feeding on them all the time. So a lot of the information that we have is more or less speculative on the aggressive mimicry, and we really need a whole lot more specific information. But I haven't been in a particularly good position to attack that problem. Did I answer your question?
Yeah,
It's
thank
hard.
you.
Do you often see hybridization between the three types in Kansas?
Hybridization, is that what you said?
Yeah, between the three types.
Oh, not between the three types, but between what I would consider, well, hybridization, if you have hybridization, breeding across okay, if I have population A and population B, and my theory is that they are two different species. If they are mating with each other, then obviously they are not a different species. Is that answering your question?
Well, I know that can occur, but then they create like infertile offspring. Have you ever really seen that, or not? Not quite.
Well, what I have been doing is, sometimes when I have two populations and the male, the flashing pattern is similar, but maybe not exactly the same, I try and cross them. And if they mate with each other, then I assume, okay, and then it's the same species. And you're talking about the hybridization where the species are different. I haven't been, well, I'm still trying to identify the species. So I wouldn't even know if it was a different species than it was, if they mated and it was a hybrid, how would I know?
Yeah, fair enough. Thank you.
I have one more question. Photo period.
Yeah.
14 to 10, you say if you take them out of the long, put them in the short, they go into diapause? There's no acclimation time, or, just take them out of a 14-10 and put them in a 10-4, and they go, whoa, time to diapause, baby. No,
it doesn't happen quite that fast. It's over a period of a month or two. But if I try, see, normally when you try and rear insects, you'll put them in a long photo period just so that they, keep on going. And, and they'll pupate as adults. Most, houseflies and all these other insects that we rear, normally we put them in a long photo period and just, leave it in one situation. With these fireflies, I have to, if I rear them under long photo period, no pupation. If I rear them under short photo period, no pupation. But it's only when I transfer them from short photo period to long photo period for spring. See, winter, as the light conditions now in spring are getting longer and longer and longer. And so that's what's triggering the development of the larvae into pupa. And so they actually have to see that change from a short photoperiod to long photoperiod.
As Lord.
And that was actually something that I didn't realize at first. Some of my early rearing, I figured I moved them into long photoperiod and just left them there, and they never would pupate. Well, now looking back on that situation, what I should have done is taken them over to short photoperiod for a while, let them get into diapause, and then move them back into the long photoperiod.
In your book, yeah, so you described some of flashing patterns of different firefly. So I wonder just the levels of variation of this flashing pattern you can find in the field. So do you think just like really every species has a different flashing pattern, and that is a indicator of different species? And is it really like consistent across Kansas at least?
Okay. My major professor, Jim Lloyd, had that as his fundamental understanding of the flashing pattern, that each species had a different male and female flash pattern, and it would be consistent and over time. And I am finding that in Photurus, there is a whole lot more variability, especially in the male flash pattern. And so it looks to me like the males may have 3, 4 different flashes that they're producing. And I'm associating some flashes as being episomatic, some flashes as being defensive, some flashes are mating. And the thing that appears to be conservative is the female response. So that means that the female part of this flash dialogue is very important. For example, we have a species here, a Photurus missouriensis, which was identified down in southwest Missouri. And it took me a long time to find that population and identify it for sure. And when I study that population down in Missouri, it produces single flashes, and the interval between the flashes is anywhere from 1 to 3 or 4 seconds. There's a lot of variability, and then the female is also an aggressive mimic. But as I move west into Kansas, I see the same firefly that responds the same way to the female, but it's doing other flashes. Like here in Heston, it produces a hitch flash, which is 2 flashes close together. The first one is kind of a short flash, and then a bigger flash. And so there's a lot of variability in the male flash, but it appears that, from what I can tell, it's the same species.
Thank you.
This is what I'm trying to decipher. It appears that in the Photurus fireflies, the female response, and therefore the decoy, is going to be much more useful in identifying the fireflies than the male flash pattern.
Larry, you're doing all this from your home since you retired. Really, really interesting. What we want you to do is get over your health problems, come here and visit us, and give us a more up-to-date seminar. Thank you very much. It's really great.
Thank you. Thank you for the opportunity to present.
There's refreshment, anybody want